Okay, so here I would like to just give you a brief introduction or overview to your immune system. Now, in order to talk about a system in the body, the first thing to understand is why it's important and how it's important. And when we think of our immune system, we of course go directly to pathogens. The immune system is important for protecting us against potential pathogens, and that's very true. There are over 1,400 known species of human pathogens. And these human pathogens are going to be a wide variety. They can be anything from a virus or a bacteria. They can be a parasite. They can be a fungus, a fungi, or in some cases, in the case of a prion, a protein, an infectious protein. So all these varieties of pathogens your immune system has to be able to respond to in order for you to survive. And the immune response to these pathogens is very diverse, from an intracellular virus. Viruses are always intracellular. And what that means is the virus replicates inside of our cells. And then this goes all the way to our parasites that can be extracellular. And these are things like worms. And they can also have intracellular parasites like malaria. And we have bacteria and fungi also that can have intracellular and extracellular types of microbes and pathogens. Now, one thing to keep in mind though, of all the microbes, this is only or less than 1% of all microbes. And so what does this mean? This means that our bodies are able to protect us from most microbes out there, or the microbes simply don't have the ability to infect us. So for human diseases, of course, we're interested in those pathogens that actually can cause disease inside of us. And so that is definitely one of the important aspects of the immune system. Now, there is another important aspect of the immune system to consider, and that's that the immune system's whole role is to maintain or help to maintain homeostasis. And homeostasis just means normal. And so what I mean by normal is that if you get a cut on your hand, if you get infected with a bacteria, if you get infected with a virus, your immune system is there to bring you back to normal. Your cut on your hand, your immune system will be involved in the repair. For the bacteria and virus infections, they will be involved in clearing those infections and then repairing everything to get back to normal. So the immune system does have a very important role just in everyday life as well. Your immune system is there to remove dead and dying cells in order that new cells can replace them and can maintain the function of your organ systems. So homeostasis and bringing things back to normal happen daily, and your immune system has a role in that. So what are some big immune concepts? So the immune system has to do several things. The first thing it has to do, it has to be able to know thyself. And this is important because we have something called autoimmunity. And if the immune system attacks self, that is autoimmunity. So to know thyself is important, and this is tolerance. And so the immune system has to know what is you in order that it doesn't attack, but it also has to be able to detect and eliminate pathogens. Now, the ability to detect pathogens and the ability to not attack yourself are tightly linked together. And so we think of our immune system being effective if it's able to protect us, but we view it as being detrimental if it attacks us and causes autoimmune diseases. The immune system also has the concept of memory. The immune system does remember things. And this is a component of your acquired immune system, which we'll talk about. But memory is that if you live in an environment and there is a common pathogen circulating in that environment, the likelihood of you getting in contact with it over and over again is very high. So the immune response wants to remember that pathogen so that it can attack that pathogen faster the next time it comes into contact with it. Now, the concept of tolerance and the concept of being able to target unwanted pathogens is this whole idea about, is your immune response your friend or is it your foe? So there is a wanted function, and that wanted function is, of course, clearing pathogens. And so this is where it is your friend. And so there are two ways, or two basic categories of your immune response. We have our innate response, and this is often referred to as the first line of defense. And then we have our acquired immune response. And this is more of a learned response. And this is what gives you memory. Now, a failure of your immune response is an unwanted function. And this is where it is your foe. And these, for more modern society, are our allergies and autoimmunity. Now you can also put in here transplant rejection, that's for modern. But these unwanted functions we'll refer to as foe. Now, in these unwanted functions, the immune system is not acting irregular. It's actually doing what it knows how to do. It's just that it's creating a response that we do not want it to create. Now, there is also a physical failure of the immune system. And this is usually in the form of an immunodeficiency. And so we have our primary immunodeficiency. And this is typically genetic. So some component of the immune response is not present. And so individuals have a hard time clearing pathogens, for example. And there is a secondary immunodeficiency. And this is usually caused by some sort of chronic infection or chronic disease, I'll call it. And so we, of course, always want our immune system to work for us, but unfortunately, our immune system can, because it's such a potent system, have some deleterious effects on us. Most common is going to be, of course, for individuals suffering from the allergic response. Now let's just talk a little bit about your innate response. Your innate response is referred to as the first line of defense. And so this is more of an ancient arm of your immune response. It is found in most organisms, from the amoeba to us. There is some form of innate immunity, and this innate immunity tends to react fast. So it is ready, it's waiting in your body for something to enter that it does not like, and then it will attack it. It is very non-specific. Now, a lot of times people get confused about what we mean by specificity versus non-specific. So in the case of the innate immune response, the immune response can detect a bacteria. It can detect a parasite, but it can't tell you what it is. It does not know if the bacteria is E. coli. It does not know if the bacteria is a staph infection. It does not know if the bacteria is a strep infection. All it knows is that it's a bacteria. It's a very broad, sweeping brush saying you are infected with some bacteria. Same thing with parasites, same thing with fungus um, and viruses. It can't tell the difference. It can't tell the difference between the cold. It can't tell the difference between the flu. And so that's where it's nonspecific. Now, some of the aspects of your first line of defense are just simply barriers. So we have our barrier function. And so the obvious one is your skin. Your skin keeps pathogens out, keeps microbes out. You also have your salivary glands. And these are going to secrete lysozymes. And these are going to be an enzyme that digests. And so just like it will digest food particles, it will digest bacteria, it'll digest different microbes. We also have our stomach. And this is going to be a low pH. So not all potential pathogens can cross into your stomach and get into your small intestine. There are a few that can. H. pylori, Helicobacter pylori, is a cause of stomach ulcers, and it can survive in the stomach. It cannot survive in the low pH. It has adapted virulence factors that allow it to sort of encase itself in a pH bubble. So that pH bubble will protect it from the stomach. But the stomach and the low pH will break down most of our microbes, will break down most or many of our pathogens. There are some, however, that have evolved to get through that barrier. We also have our normal flora. And these are typically bacteria, can be yeast or a fungi. Candida is a prime example of a yeast that colonizes us. And so our normal flora, their role is really to take up space. Their role is to make sure that a pathogenic version of them, pathogenic bacteria, cannot colonize us. So we can live with our normal flora microbes, and if we wipe them out, we have the potential to have pathogens move in. And this happens often in the hospital situation where people are on antibiotics and you wipe out the normal flora, and then you get what many people know is this Clostridium difficile infection, for example. Now let's just talk a little bit about our cells. What cells of the innate immune response is helping to provide this first line of defense? And so we have a group of cells that are called phagocytic cells. And this simply just means that they can eat things. And the nice thing is there are just a few of them, so they're not hard to remember. We have our macrophage. And these are symbolized with a circle with a line. These are tissue resident. And this is important because that means they're hanging out in your tissues. They're going to be there. They're going to be your alarm system. So when you have a cut on your finger, the macrophages are there. And when the bacteria enter that cut, the macrophages are going to be there to be alarming the immune response that, hey, a bacteria is here, come and help me fight it. Now you also have a version of macrophage that's in the blood, but we give it a different name when it's in the blood. These are monocytes. Oops. And so these are macrophages in the blood. It's the same cell, comes from the same progenitor cell. It's just once it enters the tissue, it is able to walk around and migrate through the tissue, so it's called a macrophage. And when it's in the blood, it has different receptors to help it get out of the blood, and so it's called a monocyte. And the third one is our neutrophil. Our neutrophil is going to be the most abundant cell, white blood cell in your body. This guy likes to hang out in the bone marrow. And this guy is going to be referred to as a granulocyte. Granulocyte means it has granules, and the granules are going to contain a lot of toxic things that will help to fight against bacteria and other pathogens. The neutrophil is going to generate pus. So when you have an infection, the neutrophils will leave the bone marrow in mass, go to where the macrophages are screaming there's an infection, and they will simply try to control the infection and they die. And so pus, for example, if you have pus on a finger, those are dying neutrophils. So those are our phagocytic cells. Now we also have another cell type called a natural killer cell. And this cell is a lymphocyte. And so lymphocytes are about the same size as a red blood cell. Your macrophages, neutrophils, are much larger. Now, so your natural killer cell is going to be very important, not only for pathogens, but it also has a role in homeostasis. And so your natural killer cell can induce apoptosis, which is called killing of another cell. And so what the cell is going to do is it's going to have a molecule on the surface that can recognize another molecule on a dying cell surface, and it can cause that cell to undergo apoptosis. So for example, we have our natural killer cell, and then on its surface it's going to have a molecule called Fas ligand, and then we're going to have a dying hepatocyte over here, a liver cell. And this guy on the surface will express a molecule called Fas. And so Fas, binding to Fas ligand, will cause the natural killer cell to kill that hepatocyte. And so the hepatocyte will be fragmented. And will die. And this is so that it can be replaced by a new hepatocyte. And so your liver can continue to function normally. So the natural killer cell has this ability to do this. Another cell type is called our dendritic cell. And our dendritic cell is going to be a link between adaptive and the acquired immune response. An innate response. It is not part of our adaptive immune system, our learned immune system. It is part of the innate system, but it's going to interact with our adaptive immune system and get the adaptive immune system going, which we'll discuss. And then we have another group of cells, our granulocytes. And it just means they have granules. We've talked about our neutrophils, so that's also our phagocytic cell. And our neutrophil is going to be important for things like extracellular bacteria. And then we have also our eosinophil. And this is important for parasites. And we have a basophil. And this is important for the allergic response and parasites. Now, one other cell that I'll mention here, let's squeeze them in, is our mast cell. And this is, of course, for the allergic response plus parasites. So you can see our allergic response is overlapping our parasite response. And so what basically that means is that this allergic response was developed evolutionarily to help us combat parasites. Parasites go hand in hand with sanitation. So as we increased our ability to be more sanitized and have better water systems, etc., the exposure to parasites went away. but this system is still in place and is responsible for most of or all of the allergic responses. Okay, so let's talk a little bit now about our adaptive response and the cells involved in this. And so adaptive is going to be specific. Now, while I told you that the innate immune response is nonspecific because it can recognize a bacteria but doesn't know which one it is, it's the opposite with your adaptive response. Adaptive response can tell the difference. So specificity equals one cell, one antigen. And the antigen is going to be on a pathogen. So what this means is that a T cell that recognizes influenza cannot recognize your COVID virus. A T cell that recognizes measles cannot recognize polio. If a T cell recognizes staph, it cannot recognize strep. So they're very, very specific. Now, our two different types of cells in our adaptive response are our T cells, and we also have our B cells. Now, our T cells have two different types. So our T cells are going to be our CD8 T cells, and this is our cytotoxic cells. And we also have our CD4 cells, and this is our helper cell. Now the CD is cluster of differentiation. That's what it stands for. So it's cluster of differentiation 8 and cluster of differentiation 4. We're going to see many of these CD molecules, and they're basically on the surface of our immune cells, and they help us to identify what type of immune cell that is. So when you see a CD8 on the surface of a cell, you know to call it a cytotoxic T cell, for example. So our cytotoxic T cells also have that fast ligand that we talked about with our natural killer cells, so it can kill similar to that, but it has other mechanisms to kill, and we'll discuss that more when we talk about the function of our cytotoxic T cells. Our helper T cells are really going to coordinate the immune response. So it really is our helper cells that are going to say, hey, we have a parasite here, or hey, we have a bacteria, or we have a virus. And the helper cells are really going to drive the appropriate immune response to counter that particular pathogen. Now, our B cells are going to make antibody. We'll talk a lot about our B cells, we'll talk a lot about antibodies, and our B cells are going to differentiate into plasma cells. And plasma cells are long-lived in the bone marrow. And so to give an example, your half-life of antibodies for measles is 50 years. And that means that the plasma cells that are making antibodies for measles live in your bone marrow for 50 years. You also have short-lived, and these are going to be in your secondary lymphoid tissues. Your lymph nodes, your spleen. They last days to weeks to months, but not years like our bone marrow plasma cells. And so basically how it works is you have your B cells, and this is in our secondary tissues. And this would be like your spleen, your lymph nodes. And the B cell is going to make antibodies. And our antibodies, we're going to draw them in the shape of a Y. And these B cells are specific for a little circle antigen. And so when this B cell becomes a plasma cell, this plasma cell takes on a different shape. It gets kind of big, and it has sort of a very granular looking cytoplasm. And that's because it is just packed with endoplasmic reticulum. And so this is going to become a, it's going to become a antibody production machine. But the specificity stays the same. So it's still going to release antibodies with that little Y-shaped, and they're still going to only be able to bind to those orange little circles. And so whatever antibody that B cell made or targeted and became a plasma cell, that plasma cell also must target that same antigen or pathogen. Okay, so let's talk a little bit more about B cells. So our B cells is going to have what we call a B cell receptor on its surface. And the B cell receptor essentially is like an antibody. Now the B cell receptor will have at its very end an antigen binding site, identical on both sides. And this antigen binding site will bind to the antigen. So in this case, we'll do a pink rectangle. And we'll put one over here too, pink rectangle. They're supposed to be the same. And so the B cell will get activated because of this pink rectangle. And it will send an activation signal. And it will release an antibody with the same specificity. Now let's look a little bit closer at our antibody. Antibody is the same as our B cell receptor. Nothing's changed. It still has a little cup to bind to that pink rectangle. If the pink rectangle's around, that has not changed. Now we call this B cell, we call these antibody in the sections of the antibody a little bit different. So we have this short chain here, and we have one on each side, and that is called our light chain. And you can see that it's short. And then we have a longer chain, and it kind of forms this trunk. And this is called our heavy chain. So it goes all the way to the end of your antibody. And then we also have the area where the antigen binds to, and this is referred to as our Fab region. And there's one on the other side, and this equals fragment. Antigen binds. And then down here in this trunk region, we have the Fc, and this is a fragment that crystallizes. Now what's important about the Fc region is the Fab region is specificity. So that tells you if you're binding to a cold, a flu, the measles, staph, strep, etc. And the Fc region is function. Now you guys might have heard of terms associated with your immunoglobulins such as IgM, IgG, which we'll talk about in much more detail. But the Fc region is where you really get your IgM, IgG, you get your IgE for the allergic response, and IgA. And so the function of each of these different Ig molecules is different, and that is in the trunk. It's in that Fc region. The binding to the antigen, the specificity, is in that Fab region. And that will never change regardless if the Fc region changes. So just keep that in mind. Okay, so now let's talk a little bit about our adaptive response. So we drew up there with our B cell, it was binding to that pink rectangular antigen. So if this pink rectangular antigen was floating around and we had a T cell, and the T cell is different than the B cell, so here's a T cell. The T cell on its surface has what we refer to as a T cell receptor, or a TCR. Now a T cell receptor cannot see that pink floating rectangle, and so something has to help it, and that's when your innate immune response comes along. And in particular, what's important is our dendritic cell. So here's our dendritic cell. Now our dendritic cell is going to pick up our pink rectangle, and it's going to chop it into little pieces, and these are referred to as peptides. And their dendritic cell on its surface, let me move this guy a little closer. Okay. The dendritic cell on its surface will have its own molecule, and we'll call it an MHC, because that's what it's called. And MHC stands for major histocompatibility complex. And this MHC molecule will load this peptide onto its surface, and it'll fit kind of into the groove of the T cell receptor. And this is how a T cell gets activated. A T cell must see antigen in the form of a peptide, and it must be associated with an MHC molecule. So that's how the adaptive response recognizes antigen. B cells recognize antigen directly. A B cell can see that rectangular pink box. A T cell cannot see the rectangular pink box. The T cell must see it in the form of a chopped up peptide bound to a major histocompatibility molecule, and then it will become activated. Okay, so let's just go over what we've talked about here. We've talked about innate responses and we talked about adaptive responses. So our innate responses are fast. Our innate responses are non-specific. They have no memory. They are considered the first line of defense. And they're going to help the adaptive response. The cells are going to be our phagocytic cells, our natural killer cells, our granulocytes, our dendritic cells, and our mast cells. Now keep in mind our phagocytic cells are going to be our macrophages, our monocytes, and a neutrophil. Our granulocytes are our neutrophil, our eosinophil, and our basophil. Now our adaptive immune response takes time. It needs to learn. And this is during our primary response. Primary means the first time you've ever seen the infection, the first time you've ever seen measles, the first time you've ever seen chickenpox. That's the primary response. And then it's going to be very specific. It's going to be one cell to one peptide antigen. It's going to have memory, and the memory allows it for a faster subsequent response, so secondary plus response. The next time you see measles, polio, flu, etc. So it learned. We have the different cell types. We have our T cells and we have our B cells. T cells are CD4, which are our helpers, and CD8, which are cytotoxic. We also have B cells that make antibodies. And B cells become plasma cells. So a plasma cell is not really a different cell type. A plasma cell is a stage of B cell development and growth. Okay, so that is the end of this overview. Thanks.